Hi everyone. In today's session, we are going to explore two metrics that can support hit selection. They are called ligand efficiency and lipophilic ligand efficiency. These metrics have been the subject of much debate, and that is perfectly fine. Healthy debate means people care about improving the process. Think of these rules not as strict laws but as guidelines, metrics that can highlight, in some cases, strengths and weaknesses in your compounds. Using them is not about blindly following rules, but about gaining deeper insights into your dataset and making more informed decisions. Before we dive into calculating these properties in DataWarrior, we will step back a little and revisit some key concepts in small molecule drug discovery, so you can see where these metrics fit into the bigger picture. Drug discovery is like solving a multi-dimensional puzzle. A compound that works in the test tube is just the beginning. In the end, it must also be effective, safe, and suitable for real-world use. In the early stages of drug discovery, once the target and disease have been selected, the first task is to identify, characterize, and prioritize hit compounds. At this stage, many guidelines can be applied, from the rule of 5 to ligand efficiency and beyond, but these rules should always be interpreted in the context of the project. Used wisely, these rules help focus efforts on high-quality starting points that often are easier to optimize later. This next step involves lead identification and optimization. Here again, several rules and predictive methods can be applied. This step involves lead candidate selection. This is very challenging, but overall, the key takeaway for today's topic is that all these rules and guidelines aren't laws. They are shortcuts learned from decades of experience. Tools like DataWarrior let us apply some of them efficiently, visualize trade-offs, and make informed decisions. By combining computed data, for instance, from open source packages with DataWarrior's powerful visualization and analysis capabilities, you can explore your compounds from new angles and uncover insights that might otherwise be missed. Too many rules can kill the rules. Metrics are maps, not the territory. Use them to guide the journey, but don't let them decide everything for you. Computing these rules is not a waste of time. In general, one always gets some new insights. Today we will focus on computing ligand efficiency, or LE, and lipophilic ligand efficiency, or LLE, in DataWarrior, and see how these metrics help us evaluate and prioritize molecules. Again, there has been much debate about all these rules. I won't go into those discussions here. The aim is simply to give you a sense of some common criticisms of these rules. In my work, I run all these computations and then decide whether they make sense. This is a cartoon that says, ask 10 scientists which rules should be used, and you will get 12 different answers. When reading medicinal chemistry articles about drug discovery, we see values like these for affinity, number of atoms, and so on. In general, going from a hit compound to a drug, the size of the molecule tends to increase as new groups are added. This is often to try to increase potency and to improve some other properties. On this slide, some comments about how to compute ligand efficiency. And on this slide, some comments about how to compute lipophilic ligand efficiency. This value helps identify molecules that are potent without relying on being overly greasy. This is just an example to give an idea of how to use ligand efficiency values. In general, good ligand efficiency values are typically around 0.3 per heavy atom, though this threshold depends on the specific target, the stage of drug discovery, and various other factors such as selectivity requirements and pharmacokinetics considerations. On this slide, some additional comments about lipophilic ligand efficiency values. This is just a theoretical example to get an idea about these two metrics. On this example, which was discussed in some research articles, we see a real-world example of these metrics on HIV protease inhibitors. But there are also some other considerations than ligand efficiency, such as better resistance profile of darunavir. On this slide, I discuss using these efficiency metrics with docking scores. This is inherently risky, since docking scores are not accurate measures of binding affinity, but it can still be attempted to see if useful insights about the compounds can be gained. Now we will compute these metrics on thrombin compounds and only focus on ligand efficiency values to illustrate our previous discussions. So in DataWarrior, we go to databases and write thrombin in the small window and select Homo sapiens. We will keep only molecules that have p-values, and we also select molecules with inhibition, constant values as shown on the slide. Then we compute molecular weight to remove very small fragments and molecules that are too large. Now we keep molecules with molecular weights between about 300 and 600 and delete the others. In this database, the ligand efficiency values are already present, but we will compute them again with DataWarrior. For this, we go to Chemistry and we calculate properties. Ligand efficiency is computed here from the column value. Then we can add a new 2D view by clicking on the plus sign. The default plot should be scatter plot, and we may want to adjust the size of the marker using the configure view icon and color the marker according to ligand efficiency values. On the XY icon, we want to plot molecular weight versus ligand efficiency values. You should get a plot similar to the one you see on the screen. The region in the top left corner could contain interesting compounds, thus molecules with lower molecular weights and higher ligand efficiency values. We can also click on the other side, as shown, and we should see compounds that are not so good as starting points. This is illustrated on this slide, where we see a molecule with high molecular weight and low ligand efficiency. This is just another way of looking at compounds using this plot. We can modify the plot if we click on the XY icon and select unassigned to the x-axis and ligand efficiency for the y-axis. The top compound by ligand efficiency contains a boron atom, which likely forms a reversible covalent interaction with the target. While such molecules can be valuable for certain projects, others may require non-covalent binders, making it worthwhile to examine other compounds with good ligand efficiency values. Drug discovery is very challenging, and our discussion of ligand efficiency metrics actually illustrates just a small slice of why this is the case. That's it for today. Thanks for following along. I'll be sharing more tutorials to continue our journey with DataWarrior.